The cell membrane is composed of a phospholipid bilayer. Phospholipids have a hydrophilic head and a hydrophobic tail. There are many different parts that are being used in cell membranes, such as transmembrane proteins that move polar molecules through the membrane, glycolipids and glycoproteins that serve as cell IDs, and cholesterol that maintains the cell membrane's fluidity. Simple diffusion is diffusion of small nonpolar molecules through the phospholipid bilayer unaided. When molecules go through the cell membrane with the help of a transmembrane protein, this is called facilitated diffusion. Both this type and simple diffusion don't use ATP and go with the concentration gradient. Active transport is when the molecules use ATP to go against the concentration gradient with the help of transmembrane proteins. Exocytosis is a type of bulk transport that happens in the cell. It is the opposite of endocytosis and is when the cell needs to get rid of waste. It starts when a vesicle approaches the cell membrane. The vesicle is then absorbed into the cell membrane and then expels the waste into the surrounding environment. An example of a molecule that uses exocytosis is a hormone, such as testosterone. Now we'll be going on to endocytosis. Pinocytosis and phagocytosis. Pinocytosis is the bringing in of liquids into the cell. The first step of pinocytosis is when liquids approach the cell membrane from the outside. When the liquids approach the cell membrane, they get engulfed by the cell membrane and then get pushed into the cell and form a vesicle. Phagocytosis is the opposite of pinocytosis. Although it uses many of the same steps to bring in the desired material, instead of importing liquids, phagocytosis imports solids into the cell from outside of the cell.